these bacteria are resistant to most of our commonly used antibiotics, especially third-generation cephalosporins. Now, people who carry resistant bacteria or are colonized by resistant bacteria may not be sick, but they remain at a high risk of developing resistant infections later on. What is concerning is these people, they act as silent reservoirs of these resistant bacteria, spreading them within households, hospitals, and the wider community. A study we did in 2019 showed us that 1 in every 2 persons in the community settings are colonized by ESCRE. So this goes to tell us or give us more evidence that ESCRE, previously thought as a hospital problem, is now increasingly common in the community settings as well. However, despite this increase, we still do not understand how this colonization happens over time. And this is because most studies in this area are cross-sectional, including that 2019 study we did. They are all cross-sectional, and so they do not tell us much about the dynamics of acquisition and loss of these resistant bacteria. So this study aimed at addressing this gap, and we conducted a longitudinal study assessing mothers and children to better understand how colonization with ESCRE changes over time. So we enrolled 133 households from 2 sites or 2 villages in Kibera that are sites for KEMRI CDC-sponsored population-based integrated disease surveillance. And from each household, we selected a child less than 9 months and their mother, aged 18 years and above. And we visited these pairs every 2 weeks for 12 months, constituting 24 visits. And we collected, among other data, stool samples that were taken to the lab for testing for presence of ESCRE. So we coded presence of ESCRE as colonization and absence not colonized. And then we defined long-term carriage or persistence to be continuous colonization exceeding 3 months. So here we see a heat map showing individual women, their colonization patterns. That's on the y-axis. The x-axis shows visit numbers. Like I mentioned, we had a total of 24 visits. The red tiles indicate colonization at a particular visit, and the gray tiles indicate no colonization. So what we observe is that all 133 mothers, at some point during the study, they were colonized. And if we focus, for example, on visit 1, those red tiles along visit 1 indicate to us that 75% of the women came to the study already colonized with ESCRE. The remaining 25% acquired colonization within 3 months of being in the study. So these colonization levels remained high, consistently high throughout the 24 visits, averaging between 80 to 90%. Their average colonization frequency was 16 times. So out of the 24 sampling points we had, on average, each woman was colonized 16 times of those. We also had another subset of these women, 25 in total, approximating to 19%, that were colonized at every visit where they provided a sample. So, altogether, these observations tell us two things. Colonization in this cohort was stable and it was sustained over time. If we look at children, the same kind of patterns as well come out. All 133 were colonized at some point, and their colonization frequency was 14 times. So unlike their mothers, most children came in to the study without ESCRE. So only 40% were colonized at baseline, that is at visit 1. But within 3 months of being in the study, they had already acquired ESCRE. And 21% of them, like their mothers, were colonized every time they provided a sample. So again, we see colonization here is frequent and it is recurring. So we went further to look at colonization among, like joint colonization for the mothers and children. So if the mother is colonized, is the child also colonized at that same visit? So what we found based on the red tiles, which indicate that both the mother and the child were colonized, the takeaway here is that colonization is common, the joint colonization is common. However, it doesn't persist for long, except for these 11 pairs where every time they both provided a sample, they were both colonized. We also looked at consecutive colonization. This speaks into transience and persistence of colonization, like how long are they keeping colonization. So what we found is that most children are colonized for shorter periods of time, that is 1 to 3 months, and this suggests that children clear their colonization sooner, as opposed to the adults who seem to be keeping the colonization for longer stretches of time. So based on everything we have described, what we would say is that colonization in this setting is common. It is frequently recurring, possibly driven by ongoing exposure. And so the implication is that any control measures, for them to be effective, we must integrate approaches that reduce exposure and approaches that interrupt transmission. This can be done by addressing the environmental or behavioral drivers. I'd like to acknowledge CDC for funding this study and our collaborators at KNH, KEMRI, University of Nairobi, County Government of Nairobi, and of course the Kibera participants for providing the data to us. Thank you very much.
Thank you very much, Ita, for that insightful and very precise presentation. She went slow but sure, okay? Yeah, and you spared us some few minutes. So Asante, then we move on to our last presenter for this morning session. That will be Aruse Chebet. So we shift from humans to now animals. Is it animals or environmental? She'll tell us more. Or is it the gas emissions? So Aruse will present linking antibiotic residues degradation to microbial dynamics and greenhouse gas emissions in manure. Karibu sana.
Good morning, everyone. My name is Lynette Ochieng from ILRI, and I'll be presenting this work on behalf of my colleague, Aruse Chebet, who is a graduate fellow at ILRI. So this work is on, it's linking antibiotic residues and microbial community, and also greenhouse gas emission in manure. So a study done in Kenya by Muloi et al. Reported that more than 90% of Kenyan livestock households use antibiotics. And so when animals receive antibiotics, there is a possibility of these antibiotics being excreted in the manure. And their presence in the manure has chances of altering the manure microbiome. So when this manure containing residues and altered microbiome is used to fertilize the soils, there are chances that the residues will impact the soil microbiome and also greenhouse gas emission. A study also reported that more than 70% of Africa's greenhouse gas emission is from livestock, and about 21% is from manure management. So why is it important to understand this dynamic? Antibiotic residues, change in microbial community, and greenhouse gas emission? Because there is a possibility of transfer of AMR genes and AMR-resistant bacteria, and also an impact on the climate. To set up this study, we collected manure from healthy animals that are grazing on natural pasture at ILRI. We homogenized the manure and then weighed 10 kilograms in plastic containers. So this was done in 8 replicates. The first set of 8 replicate containers was used as a control, and then the other sets were treated with different antibiotics. So we had tylosine at a higher concentration and lower concentration, enrofloxacin at a higher and lower concentration, and also a treatment with oxytetracycline at both high and low concentrations. So these treatments were incubated for a total of 110 days. And then we used Picaro, the first equipment there, to measure the levels of production of methane gas and nitrous oxide, carbon dioxide, and ammonia. We also measured the manure nutrient content. After the 110 days of incubation, we checked for residues of the antibiotics that we had used to spike our manure samples. And then we also extracted DNA that was later sequenced, and we identified some microbial taxa and also antibiotic-resistant genes. So for the fate of antibiotic residues in the manure, we realized that tylosine had a reduction of 99% after the 110 days of incubation, and that's shown in the first graph. You can see our starting concentration for the high was 700, and after 110 days of incubation, there was like a very rapid reduction. For enrofloxacin in the middle graph, there was an 83% of reduction, and oxytetracycline, the last graph, a 73% reduction. So this shows that even after incubation, the antibiotics, they do not disappear in our manure, they still exist. Now, what's the effect of these residues that persist in our manure? What's the effect on microbial communities? So my figure A, it shows changes of microbial community in day 43 and day 110, measured using the Shannon index. And for day 43, we saw a lower median and a higher variability as shown by the dots. They are spaced a bit. But on day 110, we saw a higher Shannon index and also clustering of the microbial community. So when we performed Wilcoxon's test, we realized that there was a significant increase in microbial community as the days proceeded. Figure B is a principal coordinate of analysis for the overall microbial composition for the 2 days. That's day 43, shown by the dots, and the triangles are day 110. And then the colors there show different treatments. So for the spots, the spots in figure B, they show the sample microbiome, and their closeness is an indication of similarity between the microbiome. It's very clear from the circles, that's day 3, the distance between day 3 and the triangles, we can clearly see that it's wide. So that's an indication that time is a major factor in microbial community diversity. And with the treatments, that's the colors, at 110 days, you can clearly see that there was a lot of clustering in the microbiome. So that's an indication that the microbial community can persist even in the presence of very low concentrations of the antibiotics. The next was a microbial community in manure based on the taxa. And at day 43 still, there is no clear pattern. Every treatment expressed a different microbial profile. But, with the control having most of the bifidobacterium. But then at day 110, we see a characteristic microbial profile, and there is a change with an enrichment of methanogens. So the methanogens are the gray and black bars, the ones that I'm showing. In day 43, they were less, but then at day 110, the number increased. So again, what does that tell us? That when you keep your manure, there are chances that the microbiome, they're able to regroup, and there are chances that we might have new taxa that will influence greenhouse gas emission, as I will show in the next slides. So we also checked on antibiotic-resistant genes, and then we realized that the control had the highest ARGs that you can see. Even the x-axis, the values are a bit higher. And then of interest again is that aminoglycosides and also tetracyclines were the most widespread. And this shows historic use, not just recent use. It shows that these antibiotics have been used for a longer period. Of interest again is we were able to detect beta-lactam genes, vancomycin, and aminoglycoside genes. So these are antibiotics that are commonly used in the human sector and not in cattle. So the take home from this slide will be, even when you treat your manure, the residues can impact your ARGs, and again, your control sample can also act as a reservoir of ARGs that can be transferred to humans and also to livestock and again to the nearby environment. That figure shows antibiotic residues in manure and now their impact on greenhouse gas emissions. So for figure A, it's for methane. So the Intergovernmental Panel on Climate Change has a threshold of 4.4 for methane. But when you look at this graph, for the control, that's the first bar, the levels were lower than the intergovernmental standard. But for other treatments, that's enrofloxacin, both the oxytetracycline high and low treatments, we noticed an increase in methane gas production. And for nitrous oxide, for the control again, the levels were low, but for tyrosine and enrofloxacin high treatments, we reported higher levels of nitrous oxide. To conclude on this work, we would report that methane and nitrous gas emissions from manure exceeded the intergovernment thresholds, as we've seen, and this poses a risk to the climate, to the environment, and also a possible path for transmission of AMR to either livestock, to the environment, or even to the humans. And then degradation of the antibiotics varies, as we've seen. Tyrosine degraded faster than the rest. And this could be influenced by the different chemical properties of these antimicrobials and also the microbial activity in the manure. And then lastly, the antimicrobial resistant gene abundance and carbon and nitrogen cycling can be influenced by these microbials that are also impacted by the presence of antibiotic residues in our manure samples. So this work has been it's due for submission for publication. Special thanks to Arusei's supervisors at ILRI, at University of Eldoret and Moi, to her lab team, our bioinformatician Gildas, and to the funders. Thank you.
Thank you very much, Lynette, for that great presentation. Someone would think you are really involved in the work as much as you're presenting for Chebet. So I'd like to welcome all the presenters to the front so that we can head on to the Q&A session. Our online attendees, please share your questions in the chat box also. James, Simon, Ita, Lynette. So I hope you are noting all the questions for our presenters. We'll have mics going round, so if you have a question, please raise your hand. Questions for our presenters? Yes.
Can you raise your hand? So thank you for a good presentation. So my question goes to Lynette about that presentation, and is a very good one and very good research. But now my question is, you see, as we go forward, we normally discourage the use of chemicals, especially in farming. So what is being encouraged is organic farming. where now we use that manure. But then I found that even after treatment, there are still residues. So what should be the way forward? Or in short, how safe can we be sure that the manure is fine for use for the sake of the future? So what should be the way forward out of that research?
Okay, thank you to the presenters for your presentations. I have a question for the first presenter. You saw quite a bit of resistance in Kiambu, so my question was, what type of mixed farming did you see in farms where resistance was high? And then, I think should be Teresia. So it's interesting that you see shorter carriage in children compared to their moms. So what I wanted to know is, what do you think is influencing that? And are you guys looking at microbiome effect, continuous exposure from the environment? And do you think that could be influencing the carriage that you're seeing?
We can take one more question.
Mine is not a question, but it's more of just a thinking on the presenter who did on aquaculture. So you did the study in Nairobi, Kiambu, and Machakos, and the lake region also is a very high consumer of fish. I think we should also consider that region should also have been considered in this study, so that you get like a wider coverage. Thank you.
So let's get one last one from Susan.
Good morning, all.
Morning.
Mine is a comment, not a question. Yeah, since we came here on Tuesday, we've really had beautiful presentations with a lot of information. Our NAP objective number 6 is on research. And we usually say we've not done much on that objective, but from the work I've seen since Tuesday, we've done a lot of research. My challenge is, you are doing very good presentations, very informative work. Are you publishing this work? Because when we go to, when we are looking at the researches which have been done, you find in Kenya, it's not so much. But when you go to other countries, globally, you find so many researches which have been published. So it's a challenge. The good work that you are doing, plan to publish so that even outside there, we'll be seeing we are doing something on AMR. Thank you.
Thank you, Susan. So I hope the researchers in the house are publishing, okay? Yeah, so we'll start with Lynette.
Thank you for the question. So if I would answer, I think there is need to reduce on antimicrobial use. And again, I'll emphasize on having proper biosecurity measures and vaccinations where possible, because this will all contribute to lesser usage of antimicrobials. But in cases where our animals are treated, then there is need for proper treatment of manure. Yeah.
Thank you, Lynette. Eta?
No, we are not doing any microbiome studies. and further analysis as to why we're seeing children decolonizing sooner are underway. However, I think we can hypothesize, but based on our data, we cannot confirm this, that adults' gut microbiome is fully developed, and so if they get something into their gut, it will tend to stay longer as opposed to children. And as you could see, our cohort is quite young, so things are changing, things are forming, etc. But further analysis as to why children, based on the risk factors we looked at, to see what among those risk factors could contribute to the patterns that we saw is underway. And yes, we usually publish our work.
Yeah, just following up on that, Ita, I don't know if from the social dynamics bit of it, was there kind of relationship between what you are seeing, this persistence in children, the short persistence in children as compared to adults, in reference to maybe behavioral factors or environmental factors? Did you do that kind of comparison?
Not yet,
Okay.
but our previous studies, the cross-sectional study I mentioned, based on that study, one of the risk factors that really came up in association with ESR recolonization was frequency of contact with health facilities and exposure to poultry. So in fact, that bit about contact with health facilities is what triggered now the study we are doing, because we want to know how does colonization maybe affect how often you go to the hospital and vice versa, like what's that directionality there. And then, of course, other studies have shown that wash conditions play a major role in driving colonization.
Okay. That's interesting, because maybe for the kids who are visiting the hospital frequently, you'd find maybe they are put on antibiotics, so it tends to clear the colonizers. So that's why maybe you get that persistence clearing very early in children as compared to adults.
Could be.
Yeah,
I
could
hope
be.
our risk factor analysis will shed more light to that.
Okay. Yeah. One last question for you from me is, for these children, or even the mothers that were shedding the pathogens, right, or the colonizers, was there any intervention? Because remember, they still pose a risk to their household members or even to the community. So they'll still shed, and the other kids in the community or the mothers will still pick that up, and it will still spread. Was there any intervention in terms of treatment given to them?
No, there was no intervention in terms of colonization. The only thing we did, because for the mothers we were looking at possible symptoms of UTI, we would recommend that they visit the Tabitha Clinic in Kibera, but we did not intervene in what we were seeing.
Okay, great. So we look forward to more interesting results from the ongoing study. So, James?
Thank you. So there's this one question on Kiambu, where the resistance probably was higher compared with others. And the question was, which integrated farming are they carrying out? So Kiambu, first we can check on the topography. We realize that most farmers have small farms. That may be another reason why the resistance may be, because the cow sheds are probably sometime near the ponds. And then we realize that in Kiambu, the farmers do a lot of dairy farming and poultry farming. Probably they may be using a lot of antibiotics, which sometimes during the runoff that may happen. But this being a pilot project, we need to escalate this further so that we can do several work on the same region and expound to see that what these initial results are taking us through, so that a lot of information which is lacking can be integrated using a One Health approach. Because as we look forward, the One Health approach, it has the animal, both the terrestrial and aquatic. So, and there's a lot of encouragement, especially to communities, to embrace aquaculture as a way of getting additional income or a source of protein. And that's where you find Kiambu currently, there's a lot of farmers. And we know formerly, people from that region they were not taking a lot of fish, but currently sometimes they are taking the initiative and becoming a delicacy. So I think that hopefully I have answered you well, but we need a lot of work on the aquaculture sector. And then there's this comment on the lake region. We have 3 labs. You have another lab in Kisumu, and you have another one in Mombasa because of the oceans, and the Lake Victoria region because there's a production of fish, both the fin fish and the shellfish, like the oysters and others. So we started the issue of antimicrobial resistance in the Nairobi lab, but other lab moving forward, they'll be starting all those initiatives, because looking forward, if we can get funds, we'll be able to do these surveillances, because as per se, we were being funded by only our institution. Well, that may have a limitation of the capacity on our data and the amount of data we can generate on the issue of surveillance. So, and then there's another comment about publishing. We have realized we have a lot of data in our black books in our laboratory, but when you have all that data there, nobody knows about what we are doing. So I concur with you. We need to move forward and publish this work, because I know when you're working on a greater institution like the government institution, you don't need a lot of ethical approval for you to publish all this work. It's just to engage ourselves and try to write a lot on this data, so that AMR is a global issue and is supposed to be handled in a global perspective. And that's why without information, we'll not know what is happening. And the globe is a global village. We can get resistance from one region to another. So looking forward, let us share our information outside our laboratories. Thank you.
Thank you. Thank you so much, James. I think that's really very encouraging, and I'm happy at least you're doing a lot of capacity building, especially in terms of genomic surveillance in aquaculture, and that can open up, a lot of inciting information like the issue of manure, so we can, look at all that cycle, how, all these factors are affecting each other. I have one last question for Simon. So Simon, from your data, you presented, you showed us that able money was the most predominant, though I didn't see the AST results. Maybe you can just shed some light in terms of the resistant pattern for able money being a very critical pathogen. And then also, in terms of the AST data that you presented, you had TEM and CTXM, and we didn't get to see the carbapenemeses, the OXA, the NDM, and etc. So I don't know if you just decided not to show us, considering it was the main thing of the main objective of your study. Yeah.
Okay, thank you so much for the questions. So first, about the Acinetobacter baumannii being the most prevalent pathogen, we did the AST to carbapenems and beta-lactams, and I had a significant resistance. And the reason why I haven't indicated the results on this presentation is that it is on another separate presentation that needs to be published. And again, about the other genes like OXA, we've still done the molecular surveillance. And yeah, we have those genes, and we are as well going to publish them soon.
Okay, good
Thank
to
you.
know that there's still follow-up studies on that. So any more questions? Do we have any online? Okay. Any more questions for our presenters? Yes, please.
Okay, mine is a comment, just adding to what Simon said, because we've been working together. For A. baumannii, after sequencing, we were able to get carbapenem conferring genes, and those were the oxalinases. For example, OXA 507, OXA 23. And then for the phenotypic results, they were some of them were pan-resistant, actually, not susceptible to any antibiotic.
Okay, okay. Any more comments? Oh, we have questions on that side.
Thanks for the presenters. My name is Sheila. I have a question to, I think, Ita, who talked about the colonization
Sheila,
aspects
please
in
speak up. We can't hear you.
colonization in terms of the mothers or the children. My question, my well thinking is, of course, you said after 3 months, the colonization tend to wane off. But there are some instances whereby you can have children who are very, very low immunity, that then these colonizers would kind of cross the barrier and probably maybe cause maybe blood infection or upper respiratory infections. Did you go ahead maybe to follow up and see what number of children kind of gets infections, so that at the end of the day, we would want to say, for example, out of the 20 children, for example, that you followed, probably maybe 3 or 4 of those children kind of get infection, then that would inform decisions in that when you're seeing such kind of colonization, every time you can actually suggest that I'm seeing this number and I expect kind of this number of children would get an infection. So I don't know whether you went through that process. Thank you.
Good morning once again. Mine is to thank the presenters for the fruitful information and the interesting insights we are getting as far as the research you are carrying out outside here. My specific comment go to the fisheries, representative from the fisheries sector. We are seeing some interesting profiles and interesting results from the fisheries sectors, and I believe we've been working together and training in some platform regarding residue analysis. He knows very well we've been participating in some VMD training, and I believe from here we can chart the way forward of working together, fisheries and the veterinary department, so that we can have our data, do a comparison data and see how we can do more research as far as the livestock sector and the fisheries are concerned. Thank you very much.
Any other question?
Okay, Ita.
Thank you, Sheila, for your question. And no, we did not follow them up to check any infection. Our interest was purely colonization and what may be driving this colonization.
Yeah, I think I don't have a question so far, but there was a comment from Dr. Omondi. I think it's high time we work not in silos and work as multi-sectoral approach, because I realize like in developed countries, they work as multi-sector with one approach and objective, because as we look on the issue, like the example of UK, the livestock, the fisheries, and the veterinary medicine directorate work together because all these issues are interrelated. And looking forward, we have a lot to work, because when you come to surveillance, there is surveillance on aquatic disease. So these are diseases which infect our fish, where the veterinary take the lead because they deviates the competent authority on issues on diseases of all animals, both terrestrial and aquatic. Thank you.
Thank you. So I hope I've not closed anyone out. Okay, great. So thank you very much, our presenters, for the very insightful presentation. So basically, just in summary, the study on colonization, very interesting with the red tiling and all that. So we need to do a lot of work, not just in the infected individuals, but also let's crossover and also look at the role of courage of these AMR pathogens. The other is, of course, a lot of emphasis on genomic surveillance, which James has brought it out very well. So we need to do a lot of capacity building and bring all these sectors that are doing AMR work together. And then lastly is on the presence of these antibiotic residues, not just in what we consume, but also the end products from animals like manure. So it doesn't just affect the environment, but also has a very key impact on what we consume from farm to fork. So thank you, presenters, and I know I'm standing between you and your chai, so I'd like to welcome you for your tea break. Thank you. So we meet at 11 for the next session. Round of applause for our presenters. Asanteni sana.
So welcome, Victor. You can take us through.
Good morning, everyone. God is good all the time. My good name is Victor Odira. I'm a recent graduate from Jomo Kenyatta University of Agriculture and Technology, and at the moment I work with the non-governmental organization here in Nairobi known as Health Rights Advocacy Forum in the field of advocacy, policy, and governance in health. So I'm here to present an initiative, an awareness that we conducted as, in JQuat. So it was about building a culture of antimicrobial stewardship among university students. And this one was a peer-led awareness initiative, just as I said, conducted in JQuat. So it's much related to what some of the presenters did yesterday about youth and awareness. So without wasting much of your time, I just go to my introduction. It's just a repetition of what most of the previous presenters had said. So, but I'll remind you that AMR is still a widely recognized as one of the most urgent global health threats, and it is driven largely by misuse and overuse of antibiotics, and these happen in both community and clinical settings. And it is the same even in Kenya. So in university, students, we targeted particularly those in the College of Health Science and Pure and Applied Sciences, because the reason for this, we identified this one is a critical group, since these are our future prescribers, researchers, and even policy influencers. So despite all this, you find that awareness of AMR is still limited among these young adults. So this is the reasons why they were our target. So this one was a peer influence plays a major role in shaping health behaviors among young adults. That is why we did this using a peer-led model. So because you find that most of the young or the Gen Zs or the young people, we understand ourselves more. So that is the reason we decided to use this kind of initiative. So we had three major objectives for this kind of campaign that we conducted. One, we wanted to assess the changes in the students' knowledge on appropriate antibiotic use, where we wanted to know how the students understand and when antibiotics are needed, the safe use, the basic stewardship principle, and all that. Wanted also to evaluate the shifts in attitude towards antimicrobial resistance, and lastly, we wanted to examine changes in the self-reported behaviors, because we identified there's some of the behaviors that happens among the students that seems actually right or normal, but it's actually wrong in terms of AMR, in terms of antibiotic use. So we used 4 intervention activities to conduct this campaign. One, we had a peer-led awareness session where we had brief and interactive discussion where we conducted to introduce this concept, because you find that not all the students were well conversant with these terms, what it entails, and all that. So we also used the social media engagement where we used WhatsApp, because this one is one of the social media platform that is very active within the campus. It's like almost 100% of the students actively use WhatsApp. So we also engage discussion and digital campaign to reach these students outside the class setting. We also had the educational posters where these are just the clear visual materials that were displayed across the campus, specifically in the colleges that we targeted, which is the College of Health Sciences and the College of Pure and Applied Science. So they highlighted the correct antibiotic practices and the common misconceptions towards it. So we also had some short quizzes used to reinforce messages and assess quick learning during the campaign. So the campaign overview, just as I said, this one is the initiative or a campaign that we conducted within JQuat, and it was between January to May 2024, and this one allowed repeated engagement and sustained messaging. So we approximately engaged 1,200 students, and just as I said when I was beginning, we used a peer-led model where us within ourselves, we were trying to guide awareness activity, facilitating this discussion and reinforcing responsible antibiotic practices. Because you find that when we interact within ourselves, there's some of the behaviors or the attitude that we have that we can only easily communicate among ourselves. So in terms of study design and data collection, there was a pre-post evaluation using a structured survey administered before and again after the campaign to assess knowledge, attitude, and the self-reported antibiotic use. And then there was a data analysis by where the pre and post intervention results were compared to evaluate the changes in the knowledge, attitude, and behaviors. So moving forward in the result, you realize after the campaign where we passed all these messages, you realize when you compare the kind of information the students were giving before the campaign, you realize there was a change. Because when we were beginning, it's almost only 54% of the students that we interacted with had a clear understanding of what antibiotic use entails. And later you realize after the campaign, most of them understood the clear and the knowledge that they need to have about this kind of information. So you realize also the students showed a better understanding of bacterial versus viral infections and appropriate antibiotic use. One example that we realized even as we were interacting with most of the students, we realized there was this kind of behavior where you see like in the university, the student can just access the treatment and all that in the university hospital. When you just have the student ID without any kind of payment, you find that there's this kind of tendency whereby once you feel better for the situation, for the kind of the condition you are suffering from, you can use maybe the drugs once or twice and then stop using that kind of, you stop using the drugs in the name of you are well. And at the same time, you find that in the sharing of drugs whereby maybe one used the drug, then stopped using them. So maybe there's a roommate who is maybe also facing the same problem later. Then the person will be like, maybe ulikuwa na homa juzi, what did you use? So you will just tell him, Nilienda, I went to the hospital, get this kind of medication. Then he will be like, you can also help me with that drug, I think I can use it. There's no need to go to the hospital. Then it seems normal actually, but it is a wrong way. It's a wrong way because that is like a non-prescription of antibiotic behavior. So you realize after this campaign, the non-prescription of antibiotic use decreased. Most of the students were not they were not self-medicating, something like that. So other improvements include more students committed to seeking professional guidance before taking antibiotics. And this is the peer-led approach was described by participants as relatable and motivating because we were using the more relatable terms, not something that is technical that we can't understand, like something that is just normal. In attitude change, we realized also the students reported greater awareness on AMR risk, increasing agreements on the importance of completing prescribed antibiotics prescription, and there was also positive shift in willingness to educate the peers about the antibiotic use, because as much as we never had that long time to create this kind of awareness, our aim was to create something like a norm that should be going on. It was like not like a one-time awareness. So moving forward in my discussion, I have four major points to discuss on. And one, the peer-led approach enhanced knowledge by making the content relatable and easier to absorb. And notable reduction in non-prescription antibiotic use shows a real behavior change. And in terms of attitude, improved attitude suggests that stronger long-term stewardship culture, because that one was our aim, so that this thing can be something that is being carried onwards, not just a one-time awareness session or activity. Then peer delivery increased trust, openness, engagement, because there are some of the behaviors that maybe the peers can be doing in terms of using antibiotics and all that we can easily tell ourselves without maybe involving someone professional and all that. So in conclusion, engaging university students through peer-driven awareness program is an effective, low-cost strategy to promote responsible antimicrobial use. You realize the student responded strongly to relatable and repeated engagement, and integrating a similar initiative into higher education setting can contribute to sustain AMR prevention in line with the Kenya's National Action Plan. We realize that I understand that most of the university or colleges have almost the similar initiative, but not all. But so if you engage this kind of awareness session within campuses, it not only helps the students themselves, but you know, if the students have that kind of information, even in the community, in the with the parents when he or she is at home, he can actually create this kind of awareness. He can actually tell them what is wrong to use, when it's wrong to use certain kind of antibiotics, and the issue of sharing and all that, not completing the prescription and all that. So in my recommendations, one, to integrate this peer-led AMR awareness programs into the university health promotion strategies, to strengthen student training on antimicrobial stewardship, and to sustain behavior and attitude gains, and lastly, to expand similar campaigns to other colleges and institutions and support periodic monitoring and track long-term impact. I want to acknowledge Health Rights Advocacy Forum for enabling me to conduct this presentation today, and Jomo Kenyatta University for enabling us to conduct this kind of awareness in the institution. And thank you all for listening to me, and that is the end of my presentation.
And thank you so much. Quite an insightful presentation there on how peer-led delivery or AMR awareness can actually help promote better awareness within our tertiary level institutions. So we'll proceed to the next presentation, and this will be by Charity. So Charity will present on peptabolis from Trichoderma species as an emerging an antifungal agent, and this will be a mini review. And this is quite an important presentation because, as we are seeing currently, we're also starting to have high levels of antifungal resistance.
Good morning. My name is Charity Liavuli Akwea, and I'm presenting on translation of trichoderma-derived peptibles into antifungal resistance, emerging strategies that can be used to tackle AMR as a biocontrol method. How many of us have heard of trichoderma? Wow, nice. So trichoderma, oh, by the way, I'm in the context of antifungal resistance. We have been talking mostly around antibacterials and all that. I like to a little bit move towards the side of fungi. So trichoderma is generally a beneficial fungi that colonizes plants, roots, and also the stems. It has been used in agriculture mostly to tackle infection, whereby agricultural people use it to help fight harmful fungi such as Fusarium and some strains of aspergillus like Aspergillus fumigatus that mostly colonizes our maize. So we find most of the time we have reports of maize that is infected with aflatoxin, peanut butter. But to tackle that, agricultural people have been using this trichoderma, and a lot of research has been done. But now, we want to use this potential that trichoderma metabolites are presenting in the agriculture to translate into human health. So because of that, this is my presentation. So in our medical field, we have different organisms that are now becoming problematic in terms of AMR, the leading one being Candida auris. Candida auris has been reported to show multiple drug resistance to the available commercially antifungal agents, together with aspergillus. So they have been listed at the WHO priority pathogens, and we only have a few drug classes that can be used currently, like azoles, polines, and echinocandins for treatment purposes, but their therapeutic options are dwindling, and we urgently need methods of scaffolding this. So natural products, especially fungal metabolites, have been underexplored, especially in Kenya. And this is a trichoderma colonized in sorghum. This is millet, sorry. You can find that these black things that you see, almost black-green, these are trichoderma. They are filamentous fungi widely used in agriculture as a biocontrol agent. They produce secondary metabolites such as peptibles, gliotoxin, and such like. And we want to hypothesize that these trichoderma-derived metabolites could also help in tackling antifungal resistance in Kenya today. Our approach of this mini review is just to summarize that trichoderma active metabolites contain antifungal and antibacterial potential. When you compare them as the way they have been presented in the agricultural setup, and then we translate this to human health, we might be able to bridge this gap and also help in AFR innovation in Africa. How does trichoderma work? Trichoderma, for example, in aspergillus, it works by mycoparasitism, whereby it does hyphal coiling of the aspergillus, then it gets derived of nutrients, and then it dies off. Also, we have reports of lytic enzymes from trichoderma, like chitinases and beta-1 and 2,3 glucanases, have been shown potential against Candida albicans in other studies. Also, the secondary metabolites, such as the peptibles, have been explored. Uric acid. This peptibole works by insertion into a lipid bilayer, forming voltage-dependent ion channels. This will cause an osmotic balance and eventually result to cell death. Such tests have been done with species of trichoderma like Hazanium, T. longibrachia, and T. viride, and it has been shown to show ROS effect. So when we compare this trichoderma in terms of our mini review to the available antifungal drugs that we have today, like for example in the azoles, we all know how azoles work by inhibiting the lanosterol. Okay, they cause the resistance. No, but now they are showing resistance as a result of the efflux mutation, the polyeins that targeted the ergosterol membrane, the echinocandins that also inhibit the cell wall. But now for trichoderma, these peptibles, if they are to be exploited, the lipid bilayer, it will be very good because it will form ion channels that will be able to penetrate this fungus that we are having and be able to cause a leakage and eventually lead to cell death. However, we have limited studies, especially in Africa, particularly Kenya. So if we can be able to collaborate and see how this can be explored experimentally, like how it has been done elsewhere, like in this slide we have a report of evidence of how this trichoderma has been effective to tackle fungal infections. It can be so good. So I'm challenging us to think around this. We are looking for collaboration. I'm trying to write a mini review of this, but we have, in this conference, it is a One Health approach. We are having people from all fields. So ideas are welcomed to look at this. This is an example of the mechanism of action of peptibles, how it's causing the leakage and the cell death. So if we translate this biocontrol mechanism into AFR biotechnology, we find that we can be able to innovate new antifungal formulas that can work synergistically with the available antifungals to tackle AMR. So in the Kenyan context, we have different regions such as Busia, Kitui, that plant a lot of sorghum. And this is a substrate that can be used to help tackle this. How is it now brought into food? Trichoderma, we've said it's just a colonizer of plants, right? But if we want to get more of this, in agricultural setup, it is found in contaminants and it causes massive losses. So if these residues from these grains can be valorized as low-cost fermentation substrates of trichoderma metabolites, and then we do solid-state fermentation of the husks of the millet bran, it can be able to create a scalable, low-cost, adaptable to Kenyan laboratories, which will enable circular bioeconomy aligned with Kenya AMR plan. So this is just a summary of some of the findings done elsewhere on the potential of trichoderma on different extracts of MRSA. We've seen, like for MRSA, it presented around 18 diameter of inhibition zone in a study done. We've seen this to Trichoderma pseudogoniki, had a Ross effect and also apoptosis. And for Candida albicans, it presented for T. hazenium. It had 60% growth inhibition rate. So these are just some of the findings of this mini review that we are planning to publish. We've been told if we don't publish, we risk to perish. So the advantages of this, it is novel, environmentally friendly, since trichoderma is safe to use. And then the synergetic potential with the available azoles, what it can do, and also amenable to peptide synthesis modification. We risk to also have low yield, it's slightly expensive, cytotoxicity, but all in all, it's worth to take the risk. So there's need to explore this, and we are seeking for collaboration with KEMRI, Calro, ILRI, KEPSI, and anyone who feels this can work and they can bring their input. So in conclusion, trichoderma-derived peptibles, there are promising leads against antifungal resistance. They can offer mechanistically distinct, sustainable, and locally adaptable solutions. So the agricultural biocontrol knowledge can be able to inform this medical antifungal innovation in Kenya. So what do you think? From plant microbes to medical innovation, it's possible. Thank you.
Yeah, we've been told from plant microbes to medical innovation, and there's already something done on the same, especially in the agricultural sector. So we'll proceed to the next presentation, then we'll have one more, and then we'll have a panel for the questions. And this time we'll go back to human health from Dr. Marion on point prevalence survey of antibiotic use at a county referral hospital in Nairobi County. Welcome, Dr. Marion.
Good morning, everyone. Good morning. It's after tea break. We should be a bit energetic. Anyway, my name is Marion Ongayo. I'm a clinical pharmacist at Mbagathi Hospital, and I'm also currently the antimicrobial stewardship focal person for Nairobi County. So I'll be presenting our study, which was carried out at Mbagathi Hospital. So it's titled Point Prevalence Survey on Antibiotic Use at Mbagathi County Referral Hospital. So as a background, okay, that's a photo of our administration block. It's a bit dull, but wow. Yeah, anyway. So Mbagathi County Referral Hospital is a level 5 hospital. And it is one of the referral hospitals in Nairobi County. We are located at Kibra sub-county, and our bed capacity is 392. We serve up to 1,200 patients at our outpatient department on a daily basis, and our major catchment area is the Kibera informal settlement. as an introduction, okay, this has been said in so many presentations. Antimicrobial resistance is one of the top 10 global health threats, resulting in over 1.3 million deaths annually. And the misuse and overuse of antimicrobials is accelerating the emergence and spread of AMR. So antimicrobial stewardship programs have been shown to promote the appropriate use of antimicrobials. So in our case, we carried out a point prevalence survey on antimicrobial use, and it's a systematic, structured way of collecting data on how antimicrobials are used. Data from point prevalence surveys play a major role in assessing the use of antibiotic treatment and providing information to design suitable intervention strategies for optimal antimicrobial use. So our major objective was to establish the prevalence of antibiotic use at Mbagathi County Referral Hospital. And some of our other specific objectives, we wanted to determine the prescribing patterns of antibiotics in the inpatient setting. We also wanted to determine the use of culture and antimicrobial sensitivity testing to inform antibiotic prescribing habits and to identify target areas for our antimicrobial stewardship interventions to improve the rational use of antibiotic and hence address the issue of antimicrobial resistance. Our methodology, the setting and survey site, so it took place at Mbagathi, a level 5 county referral hospital in Nairobi County with a bed capacity of 392 beds. The survey design, we adopted the modified WHO methodology for point prevalence survey on antibiotic use in hospitals. For the inclusion and exclusion criteria, all patients admitted in 9 of our wards were included into the studies. So these wards included the postnatal, the OBS-GYN ward, pediatric ward, the newborn unit, maternity ward, the male and female medical wards, the male and female surgical wards, and we included all patients who were admitted in the ward before 8 a.m. on the day of the survey. We excluded patients admitted in the renal unit and the patients admitted after 8 a.m. on the day of the survey. For the data collection, we collected the data over a 2-day period. It was in January this year, and we used the Kobo toolbox to collect the data. So the data which was captured, there was the hospital level data, the ward level data, and the patient level data. So the hospital level data which was captured was mainly on the hospital administrative data. For the ward, we captured ward-specific data, e.g. the type of ward, the specialty of the ward, and the number of patients admitted in the ward. For the patient-level data, this is where we captured the demographic characteristics of the patients and more detailed information on the antibiotic use, among other variables. The data source was the patient's medical files, and these files were collected on the morning of the survey. Early in the morning, they were collected and taken to a central place within the ward for data extraction. So for the results, we'll start with the characteristics of our patients. So our patients, majority of them were female patients at 62%. And majority of our patients, that is 54.7%, were adult, followed by 31.7% who were neonates. I think the age group of between, it was between, I think, like less than 12 months, I think that was the least, which was at 2%. 93% of our patients were catheterized. Most of them were the peripheral catheterization. 26% of the patients had undergone surgery during the current admission, and for majority of them, around 90-something%, it was invasive surgical procedures. Okay, sorry. On the prevalence of antibiotic use, we were at 54.2%. That is 54.2% of our patients were on antibiotic. And this study was carried It was carried together with other sites. There were other Fleming Fund sites. And out of the whole group who did the study, I think we were the highest. So our prevalence was high. So on antibiotic use by prevalence in the wards, for our critical care, that is, we have a NICU, that is where we had the highest prevalence at 100%. The lowest prevalence was at our obstetrics and gynecology ward, which was at 39.3. For the medical, adult medical wards and the pediatrics, they were at around 50%. So distribution of antibiotics according to the AWARE classification, 51% of our antibiotics were the antibiotics which belong to the access category. This is below the recommended 60% by WHO for 2023. Our watch category, we were at 46.3%. For the most commonly prescribed antibiotics, ceftriaxone was the most commonly prescribed antibiotic, which is in line even with the presentations we've seen from the launch. So yeah, it's our #1 antibiotic, followed by metronidazole. And then we have our benzyl penicillin and gentamicin following. Antibiotic use by age group, we noticed that most of our antibiotic use was in the adult population, that is over 18 years. But again, most of our patients were adult patients. So when it comes to ceftriaxone, we can see majority of it is being used in the adult patients, and the metronidazole. So the highest used antibiotics are being used in the adult population. We can see in the neonates, we are using mainly the benzyl penicillin and gentamicin, ceftazidime and amikacin. So for the indications for antibiotic use, the highest indication was in the respiratory tract infections at 39.4%, followed by prophylaxis, both medical and surgical, at 21.2%. We had our bloodstream infections at 16.1%, and then our others, like for the GI, ENT, and musculoskeletal, that was low at around 1% for each. So the indication for antibiotic use was documented in 75.7% of the patient files. 36% of patients had been on an antibiotic which had been used and stopped during their current admission. Majority of the antibiotics, the route of administration was the intravenous routes, that was at 93%. The rest 7% were oral. Ceftriaxone and metronidazole were the main antibiotics which were being used for surgical prophylaxis. So ceftriaxone was around slightly over around 51%, and metronidazole was at around 46%. 89% of patients were on prophylaxis for more than a day. This one, it's indicating that we are not really doing our prophylaxis the right way. And almost all our prescribed antibiotics were in the Kenya Essential Medicine list. For the culture and AST, they had been requested in only 6.1% of our patients. So in conclusion, there was a high prevalence of antibiotic use, so it was greater than the WHO recommendation of 40% for, okay, though it's a range for the low middle income countries, it can extend up to 60%, but it was still high when we compared it to the other sites who had carried their PPSs at the same time. The proportion of access antibiotics prescribed was below the recommended, 60%. Majority of the patients were catheterized, and this, what this shows us is that we have a significant risk for infections in these patients. There was poor adherence to treatment guidelines. We could see that we were using ceftriaxone and metronidazole for surgical prophylaxis, and that's not what is in the guidelines. And we are using also a lot of ceftriaxone even for respiratory tract infections, which is not in line with the guidelines. There was low utilization of the microbiology lab for diagnostics, implying that most patients were being managed empirically. So for the recommendations, you should promote the use of treatment guidelines to guide the empiric prescribing of antibiotics. Sensitization of prescribers on the importance of culture and AST in guiding the choice of antimicrobial therapy. We should develop a policy on IV to oral switch. We saw around 93% were on IV antibiotics. Develop a policy for documentation of indication for antibiotic use. There were also cases where the diagnosis was not indicated, and also the documentation of the start, stop, and review dates. Stringent IPC measures to prevent infections in catheterized patients. So I would like to acknowledge the following. Our hospital administration, that is the Mbagathi County Referral Hospital Administration, the Fleming Fund. This survey was made possible through their support. Mbagathi Hospital AMS Committee. Yeah, thank you.
Thank you, Dr. Marion. And we'll now switch to the animal health domain, where we will hear from Naomi on quantification of antimicrobial use in semi-intensive broiler farms in Kenya using mass, dose, and count-based indicators. So, Naomi, welcome.
Good afternoon. Is it afternoon or morning? Good afternoon. My name is Naomi Kemunto. PhD fellow at the University of Copenhagen in Denmark, and Ilri, Kenya, where I'm doing my research. My title of the presentation is Antibiotic Use in Semi-Intensive Broiler Farms in Kenya, specifically looking at the patterns, quantity, and dosing behavior. So, how much antibiotics are used in animals? 2/3 of the global consumption of antibiotic is used in livestock production, and poultry is the fastest growing sector, contributing up to 40% of all the meat. And the focus is to reduce the use. But to meaningfully reduce the use, we must first know the current levels of use. And it should be noted that measuring the use in livestock and humans is very different. In humans, typically we dose per person. But in animals, looking at the images there, we have different species of different sizes in terms of weight, population affected. And therefore, in human, the antimicrobial use is expressed per body weight. And there are many ways or many metrics methods to quantify the use. These multiple metrics complement each other, providing different perspectives to the use. We have the dose-based metric, which assesses the dosing correctness and treatment duration. These include the defined daily dose, DDD, denoted as VET. This is to differentiate them from the UDD in humans. What's this? This is the manufacturer recommended dose, usually in the medicine inserts or on the medicine package, and it's good for benchmarking. Second one, we have the used daily dose, the UDD. This is the actual dose the farmers use on the farm. Like farmers go to an agrovet and the attendant writes on their package, go use 1 teaspoon in 100 liters of water. So that's that dose. Now getting the ratio of UDD, used daily dose, what the farmers use at the farm, and UDD, this could give us if the farmers use the correct dose, or overdosed or underdosed. So a ratio of 0.8 to 1.2 is regarded as correct dose. Anything above or below is an overdose or an underdose. Another metric underdose based is treatment frequency based on UDD. This gives us the theoretical treatment duration. So using the dose they used, how many days should this treatment be given? The second metric is mass-based, which is milligrams per population correction unit, gives us the overall quantity used normalized to biomass. It's a simple in terms of the details of data you collect, and it's usually used for national, regional, or global reporting. The last one is count-based, which gives us the actual treatment days or events, the number of days that treatment was done. This doesn't consider the amount, the weight, or the dose used. So looking in the low, middle income countries, using Kenya now as a case study, we found 9 published studies that have tried to quantify the use at farm level. In these studies, they've used 1 or 2 metrics, and very few studies that have used the 3 metrics that I've shown before. In Africa, closer home, we got 5 studies that have tried to quantify this at farm level. And information extracted from Tanzania, where they quantified dairy and poultry use, they did use the dose-based metrics. And in Uganda, they did it in poultry, where they used the mass-based metrics. And all these studies were cross-sectional. And the question now we ask is, are there any published studies that capture the quantitative longitudinal data, AMU data? We found none, and this is what this study was trying to answer. And our study objective was now to quantify antimicrobial use in semi-intensive broiler farms in Kenya using mass, dose, and count-based metrics. This study was conducted between March and December 2024. How we did this is we selected eligible farms, farms keeping between 200 and 2,000 birds, and they had a running flock at the start of the study. We visited the farms with a questionnaire where we needed the farmers to fill the number of day-old chicks they received. Then on a daily basis, they should include the number present, the number sick, number dead, and the medicine used. So in terms of they write the trade name of their medicine they use, the amount they use in terms of the number of teaspoons or tablespoons or mLs, the amount of water in liters they diluted, the number of chicken treated, and finally the age of the chicken. And on that study, we gave them a waste medicine bin where they deposit all the used packages in that bin. Then we visited the farms every 2 weeks to collect the filled-in questionnaires and give them new ones. Also collect the packages, the empty packages, and also measure the weight of the chicken at the time of the visit. Then this data from the questionnaire, data extracted from the packages, was entered into ODK, and then cleaning and analysis. 310 production cycles, ranging from 1 to 6 cycles per farm, were monitored across 73 study farms. These are 85% from Kiambu and 15% from Kajiado County. There were 30 different medicinal products that were used in these farms, and in most cycles, vitamins were the most used. And all these products, as you can see on the lower graph, they were used during the early stages of production. Then now the patterns of use a total of 17 antibiotics were used, belonging to 8 antibiotic classes, and the antibiotics were used in 68% of the cycles. Looking on the graph, the A-B plot, we see that in most cycles, antibiotics of up to 4 combinations were used. So in A, we see the proportion of the cycles, and down there on B, we see the combinations. So where we have a black dot and then it's connected, those were the connections that were used. Here, 81% of the antibiotics used were used in combinations. And enrofloxacin was the only antibiotic mostly used in most cycles that was used as a single agent. And then these antibiotics, like the combinations used, you see they belong to different antibiotic classes shown on the legend. Then the plot C shows the most or the commonly used antibiotics, and we see like the combination that is used the most are the commonly used, either as a combination or as a single antibiotic. So there we see that oxytetracycline, colistin, erythromycin, streptomycin are the most used in these farms. These antibiotics were used within, most of them were used, or maybe all of them were used within 8 to 10 days of production at the early stages of production. And these antibiotics were administered for about 5 to 8 days. Now looking at the quantities used, a total of 397 milligrams per population correction unit was consumed in the study, and the intensity of use varied across the antibiotic. As on the bar graph, we see this antibiotic flumequine was the most used in terms of the quantity, and neomycin was the least used in terms of quantity. And now on the other graph, the line graph, trying to see on each day across the production cycle, from day 1 to day 36, when the chicken are taken to the market, how much in terms of amount is used. We see that across the cycle, the amount remains relatively constant. It doesn't really change across. The fluctuation is so small. Then now coming at the dosing and the treatment period, and combining it with now the quantity. On the graph, you see, I'll call it points. The points there represent each of the antibiotic used. And the size of the point increases with the amount used, also the color. So the scale there shows the color changes from blue, which was the least amount, to red, which is the most. If you look at the x-axis, is where we have the ratio. Where you find the used dose, then you compare with the DDD. The ratio between 0.8 to 1.2, the green region is all the antibiotics falling on the green region means they were given at a correct dose. Anything outside there was over or underdosed. So on the right, should also be your right, the purple region, where it is more than 1.2, meaning the antibiotics falling there were overdosed, which constitute to 41% of the antibiotics. Then on the left, the pink side, is the antibiotics that were given that were underdosed, less than 0.8, which was about 12% of the antibiotics. Then now looking on the y-axis is where we look at the duration. Most of the packages we collected from the farms, their treatment duration is between 3 days to 7 days, and that's what we used. So if you look on the y-axis, that region 3 to 7 days. The antibiotics falling in there, they were given at the recommended duration, either overdosed or in the correct dose or overdosed, and that forms the 82% of the antibiotics. The exceptions there we have is flumequine. You can see it. It was used in excess and it was overdosed and given for more days. Then the sulfaquinoxalone and sulfadimidine, which was overdosed but given for less days. So the take-home message from this presentation is that there was frequent use of multi-antibiotic combinations. Early life antibiotic exposure is widespread, and there is high and heterogeneous antibiotic use, and over 50% of the antibiotics were misdosed. Finally, I'd like to acknowledge the sponsors of this study, my university and ILRI, where I am conducting this study. Special acknowledgement to my supervisors, San, Ashni, and Dishon, my colleagues, ILRI CapDev, County Veterinary of Kiambu and Kajiado, where we conducted this study, and also the farmers from these counties. Thank you.
We'll invite our speakers just to come at the front so that we now have the Q&A session. And thanks so much for that presentation. I think we can appreciate the scientific rigor behind our last presentation. So you can just raise your hand if you have a question, and then we'll bring the mic to you.
Good afternoon. My name is Felista from React Africa. My question goes to Dr. Marion, and good job to the work that you're doing at Mbagathi. I would just like to know what are some of the factors, since the adherence to the treatment guidelines was quite low. What are some of the factors that are contributing to that? Is it that they're not the recommended drugs by the treatment from the treatment guidelines are not available in the institution, or is it that the healthcare workers are not aware of what is recommended, or is it just a behavior problem from the facility? Thank you.
Good afternoon. My name is Dr. Yakub from Coast General, Mombasa. My question is actually similar to what was just been asked. I just wanted to ask Dr. Marion whether the clinicians are aware of the first-line drugs which are used for surgical prophylaxis. Like, for example, we have cefazolin, which is used as first-line. So is it available in the facility in the first place before you tell clinicians that they stick to the guidelines? Thank you.
Thank you so much for those insightful presentations. I have a question around the PPS for Mbagathi. So Dr. Marion, you've conducted a number of PPSs over the years. What would you say the major change you've been seeing over the years in prescribing patterns? And have you been recording the AMS interventions, and do you know what interventions have caused the biggest impact in changing prescribing patterns? Particularly, I know that 93% of IV was a bit of a shocker to see. So what has worked, what is also not working, so that we can see where we can support. The last presentation as well, it's really good to see some valuable data around antimicrobial use in animals. I think my question is around the vitamins used. Do we have an indication on whether there's any antibiotics within those vitamins? Because we know there are some products with multivitamins for animals that do have colistin or other antibiotics. Also, the percentage of multi-antibiotic products, that 81%, are those registered within the country? Are they actually registered as products that are allowed in the country? And then finally, the duration of antibiotic use. Does the study assume that the antibiotics that were being used are for treatment, or is it an assumption that it's for treatment? Or do we have an indication whether the farmers were using these antibiotics for either growth promotion or for prophylaxis? Thank you.
Maybe we can
take those ones.
Yeah,
yes.
Yeah.
So, Dr. Marion and Naomi.
Okay, thank you so much for the questions. So I'll start with the first question, which was on the factors contributing to the low adherence to treatment guidelines. So what we came to find out was there were a number of factors which were contributing to this. Initially, we found out that there were some prescribers who were not even aware that these guidelines were available. So there was the issue of access to the guidelines. There are those ones who are aware that they were available, but another challenge also came to the availability of the recommended antibiotics, because now you'll find like amoxicillin clavulanic acid is one of the recommended one for respiratory tract infections. But when it comes to the availability of this antibiotic in the facility, you find that it's most of the time it's, okay, a number of, it's usually out of stock. We don't usually have it in adequate quantities. So in as much as you are telling them to adhere to the guidelines, when you don't have the appropriate antibiotics, it becomes a challenge to adhering to the guidelines. So on the issue of access to the guidelines, we've been working on that. We've been carrying out sensitizations for the prescribers. We've been making them aware that the guidelines are available, and installing them in our hospital computers to make sure that they can access them. And also with the prescribing companion app, which someone had mentioned it, we've also done a sensitization on that so that they are able to access the guidelines through that. So we are hoping to see changes, but the issue of the availability of the appropriate antibiotics still remains a challenge, because now there is financial resources which are required to ensure that that happens. There is the second questions, the second question on whether the clinicians are aware of the first line for surgical prophylaxis, and if cefazolin is available at the facility. So yeah, we've been, okay, initially, I think initially they were not aware, but we've been carrying out sensitization of our prescribers and creating awareness on what the first line is for surgical prophylaxis. We've also availed cefazolin in the facility, and we've been updating the prescribers that the antibiotic is available, and we've been strongly recommending it to be used for surgical prophylaxis. So it's an ongoing effort. Though one of the things we've realized is that change is changing from the old habits is a challenge. As much as we have cefazolin, still the its uptake for use in surgical prophylaxis has been a challenge. So it's an area we are working on as the Antimicrobial Stewardship Committee. There was the third question on the major change in prescribing patterns from the previous point prevalence surveys that we've seen, that we've conducted, what interventions we've done, and what are the changes we've seen. what is working and what is not working. So when it comes to the prescribing patterns, we've noticed that even from the previous point prevalence survey we had conducted, the last one we had conducted before this one was in late 2023, there was not, the changes were not many, there were very minimum changes. And some of the challenges that we've noticed is that we have high turnover of our staff in the facilities. So you'll carry out trainings, you'll sensitize prescribers, you'll sensitize your clinical officers, your medical officers. They'll be aware of what they're supposed to do. So once you're done with that lot, after a while you realize that most of them have been transferred out of the facility. You have new ones who have no idea what has been going on and which guidelines they should be using. So you have to start all over again. So usually there are improvements. You will improve for a period of time, and then after a while you realize you've gone back. So we just have to continue training and sensitizing the prescribers, even as we get new ones, and the others are transferred out. Also the interns, we also have interns coming in and new interns coming in. So we are working towards, during the induction, we are working towards training them, sensitizing them on AMR, on the guidelines which are available and how to adhere to the guidelines. For the lab, at least we noticed a slight improvement when it came to the request for culture and AST. I think the previous PPS, we were at like 1 point something percent, slightly below 2%. This time we were at 6.1. So some of the challenges we've been having in the past, we've been having challenges with the availability of reagents when it comes to the culture and AST testing. So it has been discouraging the prescribers even for requesting for this test. But with in the past 1 year, with the support of Fleming Fund, we've had a constant supply of these reagents, and we've seen an increase in the lab requests. So we are hoping also, as one of our sustainability measures, we'll be able to have a constant supply of these reagents so that we can continue having these tests done. We were at 6.1 when we did our PPS in January. We are hoping to do another one very soon. And our workload has been increasing, so we are hoping to see a big improvement when it comes to the request for these lab tests. Thank you.
And thank you, Dr. Marion. You can pass it over to Naomi.
Hoping I got your question correctly. For the, you asked the multivitamins and the combined antibiotics, if they're in the country. So the products we collected from the farmers, most of them had, like for example, the one I presented that had up to 4 antibiotics, they still had like a list of vitamins in them. So I think yes, if that's the question you asked. And then on the why the farmers use antibiotics, I remember we were requiring farmers to fill this questionnaire for us, filling in what they use, what amount, the doses. So we didn't ask on the why they use that antibiotics, but now we can, I think we can derive from what we see if we go like at the farm level, giving the doses they give, maybe they give it at a lower dose. And as you saw, most of these farms, they give antibiotics at the start of the cycle from day 1 to about day 10. So the assumption is they're giving it for prophylaxis, but we didn't ask that question from the farms.
Okay, thank you so much, Domi and Dr. Marion. We can get a few more questions. You can just raise your hand, then we can pass the mic over to you.
Yes, so let me go first. My name is Dr. Kuria from Wasingishu. So mine is to Dr. Marion. Thank you for the good presentation that shows the good work you did in the survey. My interest was on the per se the 40, I think it was 43% of antibiotics used for reasons related to respiratory system. Did you process the data to find out maybe perhaps if there were recurrent use of antibiotics due to a recurrent infection that could probably suggest it was antimicrobial resistance? Yeah, thank you.
Thank you so much for that question. So unfortunately for this survey, we did not go into depth. We didn't go into that depth. So I think we didn't capture that information. Thank you.
My question is to Dr. Marion. Thank you for the great presentation. I think we all face the same issues of poor utilization of guidelines. I have a question because that is also a hiccup we are facing at Laikipia County. And because we started the quality improvement project from the poor utilization of guidelines.
Okay.
And we started with the outpatient and the emergency department. But we are facing now an issue that once they are maybe admitted, the prescriber actually calls the medical prescribers at the ED or the outpatient to ask, why did we not prescribe antibiotics because it was needed? And at times you're even facing the issue of, we are prescribing this because this is what works, this is what we know works. So we have realized there's an issue of culture, the prescribing, the practice culture around us as health providers. How are you addressing this? Because it's a hiccup we are really facing at our facilities. And for sure it's not unique. Okay, thank you.
Hello.
Hello, thank you, presenters. I just want to make a comment for the student fraternity. That's a very good work you are doing in terms of student community. So my thoughts are, I don't know whether we have maybe common courses that probably we could do on issues regards to AMR initially, that is during year 1. Then if that is there or it is not there, it's important we have during the entry level. Then at that point, probably we could pick up on champions who are students as well as lecturers who will then pick up these issues regarding AMR. And then now we plan towards probably having quarterly talks or lectures, not even quarterly, like semester talks on AMR throughout the process of the higher learning institutions, not only in JKUAT, but also other institutions. That way, the youth, the Gen Zs are very good in terms of passing information across and really practicing what is expected. So I think we need to encourage this group of our nation across. And then the second question to Marion, I think that has been spoken in terms of now that we've done the PPS, what could be like the quality improvement initiatives that you put in place. For example, you'd want to have the clinicians do an antibiotic timeout review, meaning that you'd want to review the patients who are on antibiotics within 72 hours. Then you can decide based on the lab results that you can actually escalate, de-escalate, or stop the usage of antibiotics altogether. And that then will still speak to another quality improvement initiative that has to be
done in the lab, because without...
We usually use, again, recommendation according to the manufacturers. And manufacturer usually conduct studies in HIC, which setting is very different from LMIC. And then the animal health professionals, they use the same recommendation, especially maybe to complete a dose and stuff like that. So how valid is it we use those recommendations, yet the setting is very different from ours here? That is not to you, but just anyone. Most probably VMD. Thank you.
Okay, thank you. We'll take a couple of online questions. Maybe the tech team, you can project for us. So we have one from Dr. Nath. And I think this is to Dr. Marion. So Dr. Nath is asking whether there have been any follow-up PPS after the initial one, and because of the WHO recommendations on 3 surveys annually. So maybe you can consider also going a notch higher and doing antimicrobial retrospective or prospective audits after analyzing your PPS data to act on the problem areas. A hybrid of PPS and audits with feedback and interventions have been shown to impact prescribing trends more positively. I think that's just a comment. There is an issue of colistin. I think this one is to Naomi. Please consider proceeding for reducing colistin to strictly targeted therapy, bearing in mind that we have run out of treatment options for multi-drug resistant gram-negative infections. So the last one, I think that's just a comment on the availability of medications. So Dr. Marion, you can take those, and maybe Naomi. And for Charity, just a question from my end. It was a very good study and quite an innovative way of how we can actually introduce more antifungal into our system, given the resistance rate we are seeing, especially for Candida auris. So when you are doing the review, was there any data that you came across in regards to regulation, safety protocols, and also how the harvesting is also done, and whether there are standardized systems that have been set on the same? Thank you. And to Victor, I think, Victor, that is very good work, especially in the tertiary at JQuat. But now, the initiative that you had, how do you sustain that peer-led initiative such that you're now finishing school and those that are coming after you continue with the same peer-led awareness initiatives? Thank you. So you can respond to those ones, and then we can have our next set of presentations. Thank you.
Okay, so I will take the first question on how we are addressing the issue of the culture of non-adherence to the guidelines. So as Laikipia mentioned, that it has been a challenge also for them. So it is equally a challenge for us, because we've realized that it is very difficult to change the current culture. People usually prescribe what they are used to or what they are familiar with. So one of the ways we've been trying to address this is by using our consultants, because what we've come to realize is usually people listen to consultants. If we talk as the AMS committee, it is just considered as something which the AMS committee talks about and advocates for. But by trying to reach the prescribers through the consultants, we are seeing a number of changes when it comes to the prescribing habits. Okay, it's not like a market change, but at least there are some small changes happening. Also, by providing data to the consultants, Like now we've developed our antibiogram in the facility. So when you go and show the consultants that, we have high resistant rates to the following antibiotics, and these are the recommended ones, they are able at least to pass on the message to the other prescribers and at least guide them in the direction they should follow when it comes to prescribing. So that we've seen it as one of the strategies which is working, though it still remains a challenge when it comes to changing the prescribing habits of the prescribers. There was the second one on the quality improvement initiatives to improve antibiotic use in the facilities. So from the PPS, we tried to identify areas which we wanted to focus on. So documentation was one of the areas, and we wanted to improve documentation. So documentation, even in terms of indicating the diagnosis in the treatment sheets, because this one does not happen. So one of the initiatives that the AMS committee is taking is revising our treatment sheet to include the diagnosis, so that it compels the prescribers to indicate what the diagnosis of the patient is. At some time back, we had introduced antibiotic treatment sheets in our newborn unit. And what we noticed is that when it comes to the newborn unit, their documentation is far much better. You'll find that the start dates are documented, the stop dates are documented. So what we want to extend this to the other wards, so that we have these antibiotic specific treatment sheets in the other wards, so that it can improve on documentation of the start, at least the start and stop dates. We've also started conducting antimicrobial stewardship ward rounds. So we have a team, an AMS team. It consists of the clinical pharmacist, the lab diagnostic focal person, our IPC coordinator, and our AMS chair, who has also specialized in infectious diseases. So we do carry out ward rounds, and at least this helps in even guiding people towards adhering to the guidelines, and even in increasing the requests for culture and AST tests. So those are some of the initiatives. We are also advocating for the allocation of funds, especially now that Fleming Fund is coming to an end. We are quite concerned that we might not be able to sustain our lab reagents. So we've already started advocating for funds, for allocation of these funds, so that we have continuous supply of these reagents, so that we can continue conducting our culture and AST tests. Yeah, so those are some of our initiatives. There was the question on a follow-up PPS. So unfortunately, after the one we conducted in January, we've not been able to conduct one this year. We were supposed to conduct one in October, but there was a go-slow, so we were not able to do that, but we are hoping to do one soon. And we are working towards, as the AMS committee, we are working towards being able to conduct our PPS without support from outside, from our outside partners. So we want it to be our own activity, which we can sustain and conduct at least twice every year. There was a comment on colistin restriction. We should restrict colistin. So I don't know if it's fortunately or unfortunately for us, it's not it's not an antibiotic we usually routinely have in stock. We usually procure it for our patients when our lab tests have shown that there's resistance to everything except the colistin. So yeah, that's it. Thank you.
Okay, thank you. Maybe we can hear from Naomi also on the issue of colistin. I think it was one of the antibiotics that featured in the study. And I think there is some new guidelines from OIE on the same. Thank you.
Okay. So as I started from the beginning is that we need to, or the focus is to reduce the use. Even the high level talks is about reducing the use. And we need to know the current level, then we we know what to reduce and how is the usage. So from evidence from the packages we collected, you find that the multiple antibiotics, like for example, colistin, does not appear just alone. It is always combined. From even like 82% of these antibiotics are combined. And in most of them, you find that the combination and then there are a list of vitamins. So the assumption is sometimes the farmer could go to an agrovet looking for a vitamin, and he will be given a vitamin. In the vitamins there are antibiotics. And this, I think, is a legal issue to be able to differentiate, like to not combine vitamins and antibiotics, also to look at the combinations that the antibiotics are combined. Do we really need all the 4 antibiotics in one for a particular treatment? So now that we have the data and this evidence, this is one of the legal issues that we could look at. And also, you're looking at the classification of colistin, especially in the human, it's classified as critically important for human medicine. But if you come for the animal side, it's not classified at the same, like it's an important antibiotic and can be used, and it's recommended to be used. So also that's something to look at. But also from what I presented, you see vitamins, yes, are used, and there are many other things to do to reduce the use. So from studies we've done before looking at the biosecurities at these farms where they rear chicken, the vaccination, could it be that if they have proper biosecurity, because it's poor from the previous study we did, and also the vaccination, like for here, 25% of the cycles is where they did vaccination. So could it be if they have proper biosecurity and do the vaccination, that could reduce the need for antibiotics? And as we see, these antibiotics are used from the beginning to about 10 days when the birds, they see that the birds are okay, they can survive. Could it be they're giving it to cause of fear of disease? And if they have proper biosecurity, they vaccinate their chicken as they are required to, then they reduce the need of antibiotics. And this is our next study that we're looking at, what interventions could be done to reduce this. Because you can't go to a farmer and tell them, now, you should not use antibiotics because it's bad, it's causing AMR. What alternative do you have for them? So like you invest in your business around, you have like 500 chicken. Do you just stop giving them antibiotics because your belief is giving them, then they will not die and you'll get your money. So what are the alternatives? So this is what we're looking at.
Thank you, Naomi, for that. We can proceed to Charity, and then Victor, you can finish for us.
Hello, thank you so much. Yes, a lot of studies have been done elsewhere in regard to Trichoderma species extraction. For Trichoderma, it's a fungi like any other, so they culture it in the lab and then they extract. So in the Kenyan context, how we were hoping to optimize this is by the use of substrates like sorghum and millet. And you can be able to, according to the studies and the methodology used elsewhere, they were using submarine fermentation to increase the quantity of the proteins that can be extracted from the Trichoderma species, which are then used for the function of antimicrobial testing. So yes, there are methods that have been used, and it's just still the microbial algorithms. And we just hope to optimize by using different substrates compared to the ones they were using there. And the last question was about if they have shown antifungal potential.
Any regulatory processes that have been put in place elsewhere, did you come across that as you were doing the review?
Yes, I agree. Those are some of the limitations that we do not have so much regulation around the use of antimicrobial peptides as an option for antimicrobial therapy because of issues like toxicities and such like, but they're still under pipeline. We need a lot of studies that are in vivo, but because of animal rules, there's not so much going on. Most of the ones that are done are in vitro. So much is to be done because this is still baby steps towards alternative antimicrobial options. Thank you.
Okay. Thank you. Victor, over to you.
Hi once again. So first I want to thank her for the recommendation that she gave about introducing AMR awareness in the lower years of study in the university, because I think it's something that is essential, because you find there are some common units almost across all the programs like HIV and AIDS. It's almost it's almost common across all the programs in the university when you are in first year. So introducing the same, I think, can be effective. So to the question about sustainability, how we can sustain the awareness, it's actually a challenge to us, because you find that like most of us who are campaigning, who campaigned towards this awareness creation, we almost left the school almost at the same time, and now we are spread maybe in a different working site. So although we still have the WhatsApp groups that we use during the campaign, we normally post messages, we normally post some of the posters that still create awareness. But so the problem is monitoring on how to know that how many of the students that we interacted with also create awareness to other students, because you understand we only did this to students in the College of Health Sciences and some of the students in the College of Pure and Applied Sciences. So it's actually a challenge, although we are trying to still use the platforms that we utilized during the campaign. Yeah, thank you.
Thanks so much to our presenters, and we can give them a hearty appreciation as they take their seats. Thanks so much. We appreciate it. So we'll proceed to the next, to another couple of presentations, and we'd like now to proceed to the One Health aspect. So Esbon, to a presentation by Esbon looking at the impact of antimicrobial stewardship behavior modification interventions on One Health practice among Northern Kenya pastoralist communities. Thank you. Apologies about that technical glitch. We'll proceed to the next presentation on adherence to IPC practices at cesarean section. So this presentation was by Mary Mudengi, but will be presented by Dr. Doreen Wekesa. So Dr. Doreen, welcome.
Good afternoon, everyone. Good afternoon. Mechoka. So I'll request all of you to stand up for a minute, just to stretch. All right, a minute is over. You can take your seats. So my name is Doreen Wekesa. I am an obstetrician and a gynecologist by specialty in Nakuru County. In the spirit of AMR and preventing antibiotic resistance, I will take you through some adherence practices we're doing in infection prevention at cesarean section. So this basically helps us reduce antibiotic overuse and misuse, and as a result, we are able to prevent antibiotic resistance. So during cesarean section, we tend to get a lot of complications thereafter, the main one being surgical site infection. And that is the reason why you'll find mothers being pumped with antibiotics just to prevent that. So, and we can see the global incidence of SSIs is 5 to 10%, with sub-Saharan Africa being among the leading, reporting a lot of cases with 11.9%. And this puts an economic burden to our countries and also to the families that take care of these women. So we are putting up a strong IPC model to prevent infection, and this is basically a three-model plan, and training has been done to most of us, and this has helped to reduce antimicrobial resistance or antibiotics use, as I will show you in our data. And evidence has shown that appropriate use of antibiotics reduces SSIs by about 50%. So we are integrating all these practices to reduce the resistance. So this study is taking place in Nakuru County, Kenya, with partners who are JPIGO and AIC Kijabe, with the Ministry of Health and the Department of Reproductive Health in Nakuru County, with Global Surgery Foundation and the Women Health Program being the main funders for the program. So Nakuru has a couple of hospitals that are quite big, level 4 and 5. So our main being the Nakuru County Teaching and being helped by the Naivasha Sub-County Hospital. And then Bahati, Molo, and Gilgil are the other small units that we are using to do the study. They are all level 4s with theater. So basically, we are navigating this by saying we have to use antibiotics safely during the CS or not at all. So we have the right patient, right drug, right time, right dose, and the right route phenomena to use. And what we were missing in this is just the safe use of antibiotics post-CS or pre-CS. And we've seen that antimicrobial resistance has been on the rise, as it has been shown in most studies presented across here. So what are we using? The first thing we adhere to is the antibiotic prophylaxis. So basically, the guidelines recommend using the first generation cephalosporins, but due to availability, we use the third generation cephalosporin, which is ceftriaxone, which is readily available. And we really have to look at the timing of administration of this drug. So we give it 30 to 60 minutes before the skin incision. And this is basically a holistic approach. So if, when we're doing the pre-op checklist, we ensure there's a part for antibiotic prophylaxis. We ensure that is documented and the time the drug is administered written down as well. And if we find that the drug was administered more than 60 minutes before, and probably there's a reason as to why the surgery has not been done in time, we can give antibiotics for that reason. And it's given as a single dose, so basically just before the skin incision, and we've seen a reduction of SSIs by about 57%. The second thing we look at is vaginal cleansing. So during preparation, so there's a tool we use, a surgical safety checklist. During signing, we look at all the complications we might have to endure or go through our preparation, if the antibiotic prophylaxis has been given. Then the vaginal cleansing is done between signing and the skin incision. Sorry. So what happens during this time is just prior to catheterization, which is done on this operating table, we do a vaginal cleanse. So basically, a swab is loaded on an ovum forceps, and it's dipped into povidone iodine. Then through the introitus, high up into the cervix, we do anti-clockwise sweep of about 30 seconds, and then after that, we do the catheterization. So this has shown there's a reduction of endometriosis by 40%, and also a reduction in surgical site infection by about 40 to 60%. And then lastly, during, just prior to draping, we do the abdominal skin preparation. So you use alcohol-based chlorhexidine, which is preferred over the iodine-based solutions. And you allow the air drying to take place for 3 minutes. So there are centers where they will use the dilute alcohol-based solution and then wipe it off with gauze and then now apply iodine. And then wipe out the iodine, then drape the patient. So the wiping out really does not help the preparation take effect in killing any microorganisms present. In centers where we do not have the alcohol-based solutions, you can use the povidone iodine to do the skin preparation. So, so how did this package begin? We had clinical training for our staff, the doctors, clinicians, the clinical officers, the nurses, theater staff, and all this staff were picked from the 5 centers where the study was going to be taking place. The training took about 2 weeks. We had stations for demonstrations, and then we had material to use during the training. And we also have something called the surgical hub, where you log in and you can continue reading on what is new, what is being relevant, what is new information. And there are quizzes you can take depending on the reading you've done. And it has a lot of information about safety and other disciplines in terms of antimicrobial prophylaxis and stewardship. And then data collection began, and we are tracking patients who have had CS deliveries. So basically, once they've been discharged home on day 7, there's an assigned person in all the 5 centers who calls the patients and just to find out how they're doing, and it's been quite productive. We have a few challenges in terms of availability, connectivity, but we are able to use the community health promoters and the CHS to be able to help us get this data. And then we have a mentorship package where part of the trainees during the clinical training was selected to be mentors. So we do reviews every month through the centers just to collect some bit of data and find out how they're doing, if all these practices are being adhered to, including scrubbing in for theater and seeing how we are taking up the packages to prevent antibiotic resistance and adhere to prophylaxis. So the implementation of the project started in July, and through September, we had the first quarter for our study. A total of 1,752 CSs were performed. So these are both elective and emergency cesarean sections. And we noticed that the adherence rate to the infection prevention bundles, the three, rose from 0, basically, which was not being documented, to about 88.2%. And this showed there was a reduced extent of antibiotic use. We, however, have exceptions to the antibiotic use. Of course, anyone with immunosuppression, we get referral-ins from other counties around, and we normally do not know what exactly has been going on, so we put them on prophylaxis as well. And patients who have had, let's say, antenatally uneventful stuff like premature rupture of membranes, we have to put them on antibiotic prophylaxis. So those are a few of the exceptions we have. And we usually give them a 3-day course, depending on the guidelines. And the tracking of the surgical site infections from the community was from 0 to 8.2% in July, and currently at 3.8%. If I can remember, in September, I think we had, one station had 2 cases of surgical site infection, and these were transfer-ins to the unit because of they were not able to get care. They had surgery done, let's say, in Kiambu, then they came back to Nakuru, so that they were captured in this data. So, in summary, so as much as a CS section is done, it presents itself as a chance to protect life and, of course, correct use of antibiotics. So with this brief study that is ongoing, it will take 3 years from 2025 all through to 2028. But from the first quarter of the data collection, proper antibiotic use is prudent, it can be done, and it can give good results. So thank you all for listening. Thank you to JPEGO, Nakuru, Global Self Surgery, and the county.
Thanks so much for that, and thank you for completing before time. We appreciate. We'll proceed to the next presentation by Bernard, and this will be looking at access pathways and practices in antimicrobial use, and these are insights from Kibera in Nairobi. So, Bernard, you can, you're welcome.
Good afternoon. I know the presentations at such times or this moment usually receive less attention, and especially when we have already received meal vouchers. But I hope we're not going to take long, and we'll finish within the schedule. My name is Bernard Nyauchi. I work for LBCT Health as in research department, sorry. What I'm going to present to you is findings that we got from a study that we undertook in Kibera. Specific locations are a village known as Raila village, with portions of the village. The study was done by LBCT Health in partnership with Gear Up, and here it is, and then funded by Fleming Fund. Part of this study has been presented here in terms of findings. Tuesday, my colleague made a presentation, and then there are a number of posters that have been projected right behind there. So the study that, the findings that I'm sharing basically targets the quantitative because we had 3 arms of this activity. There was the qualitative, and the qualitative was split into 2. And then there was a quantitative whose findings I'm going to share. And it's all about access pathways and practices in antimicrobial use in Kibera. So we take note that access to formal health care is very limited, regardless of the type of health care that people are seeking in informal settlements. And this is a challenge that we all understand. A number of discussions have talked about it here, and we continue to see such challenges. And just hinged on that, we take note again that in those informal settlements, there are weak regulations of antimicrobial distribution, which often leads to inappropriate use, what others might refer to as misuse. And then, of course, barriers to appropriate use of antimicrobials is associated or linked to antimicrobial resistance. And I will mention that the situation that we find in informal settlements is almost the same or worse than what we have in other settings which are not informal. So we set out to conduct a study whose objective was to examine patterns of access and use of antimicrobials within the community that I mentioned, Raila community in Kibera. In terms of methods, I've mentioned earlier on that this one was a mixed method study, but for this we narrowed down to the quantitative household survey, which was cross-sectional. We used stratified sampling, random sampling of households, identified 600 participants to participate in this study, 684, majority of whom were household heads. And then we used RedCap to capture data. This one was face-to-face interviews using a structured questionnaire with different components. We did our analysis in SPSS and utilized descriptive statistics. I would want to mention to you in advance that further analysis is ongoing, and we are doing advanced analysis, regressional and bivariate analysis, to understand the relationship between the access and practices factors that I'm listing here against antimicrobial resistance based on the data that we are having. And these are findings that probably we'll be sharing in future. In terms of findings, in terms of the socio-demographics, we note that 56% of our participants were female, and we had 64.8 who had completed secondary education and above. That includes tertiary. I also have to mention that there was about 38% who had primary education. And in terms of age categories, we split that into 2. We had a majority of them individuals who are aged above 18. 18 to 34. And then above 40% was just 36%. In terms of marital status, again, majority of our participants, that is 64%, were married, with about 27% being single. We went ahead and asked questions around antimicrobial use in the last 12 months, and we took note that 81% of our participants had used an antimicrobial within the household, or it could be them or another household member in the last 12 months. Then this tells us that antimicrobial use is still very high in these informal settlements. We again sought to know the sources. The key sources that were listed were pharmacies. Of course, government facilities were also listed. And when we broke down the pharmacies in terms of these participants or respondents seeking to get prescription before purchasing or not seeking, and we noted that so many individuals were not seeking prescription as they were acquiring these antimicrobials. In terms, I've just mentioned that 1/3 did not did not acquire antimicrobials with prescription, and that's a worry. Then we also had a small proportion, of course, on this question, who are using leftovers, drugs that had been used before, they carried them forward for future use. We again asked them about reasons for use of antimicrobials, and the key items or reasons that were coming up were managing fever and also managing cough. We are basically talking about antibiotics in this case, and of course the cases of the painkillers and mention it. In terms of adherence to prescribed antimicrobial courses is again an issue, where 1/3 reported that they were not adhering to the prescribed courses. And key issues or reasons why they were not basically completing the courses, the main one was that they would take drugs, but immediately they felt better, they would stop taking drugs. That was very common even among some of us who are inside here. But again, I want you to take note that there's a category who were not, who reported that they were not understanding the dangers of failing to complete taking drugs. So we are talking about lack of that information in informal settlements. And then others mentioned that they would, it was a measure, it was an economic measure that immediately they felt better, they would stop and then use that again in future. So there's a big issue with regards to sharing leftovers or leftover medicines. And you can see from the chart, 71% reported that they were sharing, they had shared leftovers before. And the reasons was mainly to save money and because it would be used again in future. In terms of disposal, again, we asked a question on how they dispose the drugs that they did not use. And we note that about 60% were throwing that in the trash. Is again an issue that is worth taking note of, because they would find their way into the ecosystem or into the environment, and then it could get consumed again by populations. But then we also took note of a number, 14%, 15%, who were not really disposing, but were keeping for future use. Yeah, so based on what I've just shared, we take note that economic and systemic barriers continue to limit antimicrobial use in informal settlements. When I talk of economic barriers, I'm thinking about household economics. Think about how much households get as income vis-a-vis, then they would want to split that to support all the factors around the household economics. We're talking about basic needs, food, shelter, but then health comes in there. But then we also have systemic barriers such as unregulated access, significant reliance on self-medication, still an issue, and then of course the policies that have not really been enforced to ensure that pharmacies prescribe, or pharmacists prescribe before they give out drugs in informal settlements. We make the following recommendations: that we are calling upon stakeholders to support the strengthening of access to affordable and supervised health care. And then we encourage the stakeholders to enforce antimicrobial stewardship measures. And these recommendations would help us achieve prevention of misuse, reduced risk of resistance, and then ultimately improve patient outcomes. Thank you.
Okay, thank you for that wonderful presentation. I know we are standing between you and lunch, but we're almost there. So we'll just have one final presentation, and it's good that we'll cap it up from a One Health perspective. So we'll invite Esbon.
Teaching and Referral Hospital, and Dharura Charity Teaching and Referral Hospital, and Dharura Charity on behalf of University Hospitals of Bristol and Weston. And so I'll be taking us through this presentation. This is a project that was made possible through the support of CW PAMS 2 and 2.5 grant, which we are still implementing. And looking at the background, we know that One Health is a growing field, and what we need to understand is that One Health seeks to implement a paradigm shift in our approach to health by balancing or taking into account the health of animals, human health, the environment, and even climate change. And we know that AMR has actually been highlighted as a global health problem. That is for both health and global security, and is a crucial component in One Health. And we know that in 2019, about 4.9 million deaths were linked to antimicrobial resistance. And when you look at locally, we find that we have quite scarce data, especially on AMR use, especially when it comes to pastoralists using antimicrobials in their animals. And we know that in this northern Kenya, we have pastoralists who have historically lived there. And so the knowledge on One Health is crucial to be able to address the issue of antimicrobial resistance. The objective of this study was to assess the knowledge, attitudes, beliefs, and practices in antimicrobial use among pastoralist communities. That is before and also after behavioral interventions. So how did we implement this quality improvement project? We started by gathering data on antimicrobial use practices among pastoralists who are based in 5 communities within Laikipia, Isiolo, and Meru counties. And these communities include Naibo in Laikipia. We have Ngaredare, Motunyi, and Manyangalo in Meru County. And we had Leparua in Isiolo County. And we used semi-structured questionnaires and focused group discussions. And so these were used for the baseline assessment, and also after the behavior modification interventions, we did a post-survey. So, and the interventions included training of 4 female elders who are to serve as AMS guardians. As we all know, when we go to these pastoralist communities, we normally have some respected female elders, and some of them were actually traditional birth attendants, and the others were community health volunteers. And for this work, we worked with communities who are supported by Lewa conservancies. And so we trained these female elders, and we also did trainings. We brought on board a veterinary doctor to just help us in training these female elders. And we also composed a song and recorded together with these elders and with the different communities. And for the results of this survey, a total of 51 pastoralists were interviewed during the pre-survey, whereas during the post-survey, 104 respondents were included. And we did use thematic analysis, and out of the data that we got, there were different themes, like use of antibiotics, where during the pre-survey, we found out that only a few participants noted stopping antibiotics according to the vet prescription. So people were not stopping antibiotics based on advice from a vet. However, you find that in a post-survey, most farmers actually acknowledged consulting the AMS guardian, who would subsequently consult the vet before the use of antibiotics, and they would actually follow some of those guidelines by the vet. The other theme was on procurement and advice. In a pre-survey, most of these farmers or pastoralists were buying from open-air markets. For those of you who are conversant with some of these areas, we normally have, like, for example, today you may have the market, open-air market in this village, tomorrow in a different village. And so the vendors of these antimicrobials would actually move from one market to the other, and they would actually carry them in pro boxes. Sometimes it's very hot, and so you are not even assured of the quality. However, in the post-survey, majority of the respondents are limited to procuring antibiotics from licensed agrovets, and we know that there they had higher chances of meeting someone who is qualified, who would actually give them the right advice. And a number of these farmers also admitted consulting the female AMS guardians prior to using antibiotics in the post-survey. In terms of use of animal products following administration of antibiotics, actually during the pre-survey or the baseline survey, like all the participants, they actually said, most of them actually acknowledged that they would eat or even consume or sell the animal products, or that is including milk, immediately after they have given, they had administered the antibiotics. And so they would not wait for the recommended period. And some even, there's one participant who actually said that even on the same day, they would actually even sell the animal, even if the animal died, or they would even consume. And others stated that they would consume animal products on the same day, but not sell. However, in the post-survey, the immediate consumption of meat and dairy products following administration of these antimicrobials without waiting for the recommended duration was also noted to decline significantly during the post-survey. And regarding AMR, in the baseline survey, the knowledge on AMR was quite scant. And you find that some participants noted that giving adamycin, which is oxytetracycline, that was intended for animal use, so they would actually use it for their own human use, either by mixing it with milk or using that powder on wounds. And there are some who actually acknowledge it using amoxicillin for their chicken to make sure that their chicken, maybe to improve like the weight of their chicken and all that. And knowledge on AMR was limited, with many participants stating that they had not had on antimicrobial resistance. And when we even tried to define further what is AMR, there was a misunderstanding between AMR and simply treatment failure. So they could not differentiate between the two. However, we had one participant that was aware that certain antibiotics were no longer effective, and the participant actually said that some drugs do not cure animals as before. And during the post-survey, we can see that the knowledge on AMR significantly increased. And during the pre-survey, actually, you can see that the only 3 participants who say they knew about AMR, they had heard through maybe the media campaigns about drug-resistant TB and all that. So when you look at this research, it actually highlights some key concepts for discussion, which has been supplemented from similar research in the region. One thing we can notice that there's a significant knowledge gap in pastoralist communities around antibiotics, leading to inappropriate practices. And then you can also see that use of antibiotics to promote growth in animals is directly increasing the risk of antimicrobial resistance. This is one paper which highlighted that some antibiotics were directly marketed for growth promotion, highlighting further challenges within the pharmaceutical industry. There are also direct links to human health that are important to consider. For example, there's a paper that is available where there's a review of antimicrobial isolates in the Isiolo region that showed that in cattle herds where antibiotic use is actually widespread, and this has led to the presence of MDR, Staph aureus, in raw milk. And of course, we know that the use of raw milk amongst these communities is quite widespread, and this could actually lead to resistant infections within human populations. And so we find that behavior modification intervention, like veterinary guidance on animal husbandry, biosecurity, and antimicrobial use, educational programs, and even review and feedback on antimicrobial use have been shown to be effective in improving antimicrobial stewardship in the context of One Health. And in conclusion, we know that antimicrobial resistance is a vitally important topic, especially when we think about One Health. And we have seen that cost-effective behavior interventions, such as training of even the elders, female elders as AMS community champions, are effective in driving meaningful change among pastoralist communities in lower and middle income countries. And I want to acknowledge CW PAM's Global Health Partnerships, which is formerly that, County Government of Laikipia, UK Aid, CPA, that is Commonwealth Pharmacists Association, Fleming Fund, and even the Rural Charity. Thank you.
So thanks so much, and with that we come to the end of the presentations. We'll have a small panel, so invite our esteemed presenters to have a seat, and if we have any question, the mic will be circulating, so you can just raise your hand and we'll bring the mic to you. So, questions? We have one at the back.
Thank you very much, our presenters, for the good presentations that you've given us. So my question goes to the last presenter. I'm just curious to know if there was a way that you were confirming the behavior change in the pastoralists, since you said that post-survey, you found that they were observing the withdrawal periods. And I'm also curious to know, where were they taking these animal products post-survey? The other one is, how did you confirm that now they were actually buying the drugs from the agrovets compared to before, where they were buying in the open air markets? Thank you.
Thank you. Maybe we can take one more from the audience.
So me, I just want to comment about the inappropriate use of antibiotics. It's just a comment. I want to say that each one of us here should be an ambassador, in the sense that people can use drugs the wrong way, either not finishing the dose, for the simple reason that they don't know the dangers. So I believe each one of us, whichever setting we are, we should be ambassadors. It's good we take advantage. I know we are outside the workplace. I know we're in churches, we're interacting with the chief brothers and all that. So whichever setting we are, I believe we should be ambassadors to tell the people how, and to educate them. So thank you. It's a comment. Thank you.
Okay, thank you so much. Have we left anyone out? Okay. In the meantime, we'll just read the online one. And this is more of from Dr. Nath. KEMSA contributes to some of these talkouts. What is recommended and not just push the antibiotics of vested interest. So I think that's a bit unclear. So there's one for Dr. Wekesa. Some schools of thought emphasize the need to administer prophylactic antibiotics in CS after cord clamping due to the lack of allergy history of the fetus with regard to beta-lactams. Consider enforcing this exception in CS surgeries where feasible. So that's more of a comment. So I think mostly in the online space we just have comments. So maybe Dr. Wekesa, one question for you. It's quite a good program, and we acknowledge that it's just starting. So maybe as you start off, what are some of the strategies that you are putting in place to ensure that there's systemic improvement, the environment becomes enabling such that even after the program ends, the same continues. Thank you. So I think we'll just take that last question at the back, then we can close it up there. Thank you.
I'm Sami from LVCT Health, and this is a question to the presenters as well as the other people in the audience, particularly those who have been dealing or doing research within the community. Now, we do know that the language that is used mostly in expressing AMR knowledge is usually technical English. I would wish to understand what was your experience trying to translate this language in terms of the tools that you are using to communicate about AMR in local languages, in Swahili, in local languages. What was your experience like, and how did you navigate the whole idea of trying to translate this knowledge and information into local languages? Thank you.
Okay, thank you. I think we'll start from Dr. Esbon, then just go that way. Yeah. To Bernard, then Dr. Kesa.
I'll start by responding to Esther, Dr. Esther. And she asked, the first question was, how did we confirm behavior change in the post-survey? Like what I said, the methodology that we were using, we were just using questionnaires and focused group discussion. So out of the responses of the respondents, we confirmed that initially, from their response, And when we compared that to the responses after the interventions, there was a difference in that. So it was just a baseline because we first wanted to understand what are the issues before maybe we can use a more scientific way of even confirming these changes. And how, I think the other question, how did we confirm that they were buying from agrovets and not open air markets during the post-survey? Again, it was from their responses. And remember, in the post-survey, we had 104 participants, and they were not necessarily the same participants that we had in the pre-survey, because we had trained AMS elders who, together with them, we went to the communities and trained them, and then during the post-survey, we just got an average of about 20 participants from every community that had an elder. And so we asked them the same questions that we had posed to them during the pre-survey. And just to comment about what maybe Sami from LVCT, and I want to agree with you. It's a big challenge to actually explain to, like for example, the communities that we were dealing with are predominantly Ma communities, and for us, some of us are not from that region. And what we were doing, we were using translators. Of course, there are those who understood Kiswahili or even English, but they were the minority. So we had to use translators. And even when, like, how many of us know what we call AMR in Swahili? And even if you hear what it's called in Swahili, how many of us would actually be able to know, even in this room, that we are referring to AMR? So that is a challenge that probably we need to think about. How do we translate to a language that people from all corners in this country are able to understand and relate to? Because most people, actually most responded from the study that we were doing, were confusing treatment failure with the AMR. Thank you.
And thank you so much. Bernard?
Thank you. I'll respond to three concerns, and I'll start with the one that had just been highlighted: inappropriate use, which affects even a number of us in this hall. Yes, indeed, is a challenge, and I would also encourage that we deeply focus on communication, so that we're able to understand the importance of appropriate use of antimicrobials. I'll respond to the second question, concern that was about definition. Thanks a lot for mentioning some context. Yeah, for us, we had the first translation in Kiswahili, and to be honest, we didn't have one word for antimicrobial. It was a whole phrase to describe antimicrobial in Kiswahili. Then when we went to do the pilot or pre-test in the community, we realized that even the Kiswahili that we were using was not very familiar in the informal settlements, where Sheng has really influenced Kiswahili. So we had to listen to them again during pre-test, regroup during debrief, and restructure that. So to respond to you, indeed, we don't have one word for antimicrobial. It has to be an entire phrase with examples so that communities are able to understand. And I tie that to my colleague Sami's query. Sami, again, I'll take it back to you because I know we have an interest in doing a discourse analysis on for those who probably are aware, there's what we refer to as critical discourse analysis, just to understand what individuals are saying in groups, for example, in focus group discussions, to understand, to unearth their understanding of these terms, especially the technical ones. It would be very useful for us that we go back again to do that discourse analysis, and then findings can help reshape translation of such technical terms like AMR and antimicrobials. Thank you.
And thanks so much, Dr. Doreen.
The question from the moderator, strategies to improve probably adherence to the IP bundles after the study is done. Number one, probably come up with SOPs, both intrapartum to handle this and even antenatally, and this should be part of our maternity and labor ward units, antenatal clinics, and theatre. And also this can be eventually be integrated into the county reproductive, maternal, newborn, adolescent and child health and nutrition policies, and probably in future roll out to the entire nation. And then locally in the county, roll out all this training and strategies to other centers. We have many units that are having the maternal and health clinics, but do not have the theatre bit, so they're level 3 and below. But you'll find they have a reproductive health specialist, or rather a reproductive health trained staff, so a clinical officer or a nurse or a medical officer, and then continuous medical education and updates on the same, just to come to terms and in tandem with what is being practiced out here and what is recommended. Yeah, so basically, probably those will be the first steps in taking this. And definitely we'll continue data collection just to see and bridge the gaps in places or in parts where we're not able to adhere to the IP bundles. And there are challenges we'll face, definitely, availability of antibiotics and at times even supplies, so that we'll have the top management being involved in this. And we can have an antimicrobial resistance-free maternity and theatre. Thank you.
And thank you so much. And thank you to all our presenters. We can give them a hearty appreciation as they take their seats. And to me and my co-moderator, Winnie, we really do appreciate for being with us along the journey today. And at this point, we'll invite our chair, Dishon, just to give a few remarks and guide us on the way forward. Dishon, if you can hear us, over to you.
Yes, I do. Are you able to hear me as well? Can you hear me? Hello?
Addition, yes, we can hear you.
Okay, sounds good. Just give me 1 second. All right, so once again, thank you so much. It's been two and a half wonderful days, and we've come to the close of this year's WAAW Symposium. It's another opportunity we've heard about all the amazing science as a country we are doing. Just a bit of background. This year we received excellent and wonderful 93 applications, and my team, we did a very open and clear peer review process where we managed to select 26 abstracts for oral talks and 20 poster presentations. Of course, balancing different considerations for discipline, age, gender, institutions, and others. And I hope you've enjoyed the high quality science over the last 2.5 days. And I think for me, it's been a wonderful roller coaster. From hearing the sobering numbers about the high levels of resistance we face as a country, both in human and animal, utility of cutting edge approaches such as genomics, machine learning, all the way to linkages between antibiotics and greenhouse gas emissions, linking two major global problems, AMR and climate change, to utility of bespoke social sciences. We've heard about how we can leverage on behavioral science and all that to change practice. And And for me, the most important thing throughout the entire 2.5 days is the voice of the young people. I think you've seen very many young people in the symposium. We had a wonderful session just for dedicated for youth. And these are the future prescribers, the future consumers of antibiotics. And I think if we can make one change would be to get our young people to do what is right. And I hope we can do that in the few months and years ahead. And all of us will agree that this has been a proper reflection of One Health, from human, animal, and environment. And we should be very proud of ourselves as a country. But I would like to challenge us that we've had all the beautiful science, we've had the challenge, and we should ask a very simple question to all of us. How do we translate this beautiful science to see change in policy and change in practice? How do we get the prescribers Marin was talking about in Bagadi and our colleague from Laikipia? How do we get doctors not prescribe antibiotics? How do we get culture change in our hospitals, in the pharmacies, in the agrovets, and also the public? And therefore, I charge all of us to be ambassadors of change to yourself, first of all, to your families, and to your community. Speak about the need for us to act. In conclusion, allow me to thank the subcommittee who have been critical in planning for this activity. And I have to read their names because I think they deserve that. So myself from ILRI, my co-chair, Christine Bindio from University of Nairobi, Frederick from AMREF, Owada Rose from VMD, Willie Mwangi from University of Nairobi, Purity Koskei from Red Cross, Lois Kibadi from Bomet County, Tanui Emmanuel from KNPHI, Moses Olum from Calvo, Mary Ndinda from KNPHI, Susan Kavai from Kemri, Eunice Omondi from DVS, Samira Nasir from Baraka HealthNet, Winnie Mutai from University of Nairobi, Cynthia Odhiambo from NASIC, Joseph Mwando from ICAP, Linus Tegwa from USCDC, Michelle Kasudi from ILRI, Susan Odera from University of Nairobi, Elijah Wekesa from WSU, Veronica Chuchu from CPA, Joseph Jungia from CPA, Felix Kimpegwa from University of Nairobi, James Wanjama Kabugo from Fisheries Service, Collins Jaguga from NASIC, Daniel Origi, who's just there from Z Institute, and Lois Kihugi from KNPHI. This wonderful team, they've done all the work. And I allow me to ask you to clap for them, if you don't mind. Thank you for doing that. They will take all the glory. As a chair, I will take all the blame. So if something didn't work out okay, if we misspelled your name or something wasn't okay, I'll take the blame, but not the committee. The committee takes the glory. I'll thank all the funders who supported this symposium.
Hi, Dishon. We lost you for a minute there. Can you hear us?
Sorry about that. I think his phone went off. So allow me to, all protocols observed, allow me to invite Dr. Emmanuel to close for us the meeting.
Good afternoon, everyone. Good afternoon. You're sounding very tired. It's been a long day or what? Yes? Yeah, sorry, I'll not take much of your time. I know a lot of us are hungry and we want to replenish our glucose. So let me take this opportunity on behalf of the entire WAAW Planning Committee to first of all thank the chair for the symposium subcommittee for a very well executed, planned, and high quality symposium. Let's appreciate the committee addition. I think the quality of the symposium is, has been improving, and this year is one of those best symposiums that we've been able to conduct as one of the World AMR Awareness Week. I think this really demonstrates the teamwork that we have been putting together in ensuring that we have a successful event. I think Dishon has really summarized the spirit of the symposium. I will not repeat what he has mentioned, but really to appreciate all those people that he has highlighted, all the way from the presenters who have been able to share highlights of their research. It is not very easy to summarize what you have been doing for many months and even years so that people are able to understand what you have been conducting at your area of influence. We really appreciate your time. The research that has been shared spanned across routine surveillance, new innovations. We saw very encouraging innovations from the youth, and we are happy that they are taking increasing influence in terms of AMR awareness, and we are hoping that this is going to continue. Mine is to urge, as we come to the close of this symposium, that we carry along the knowledge. We translate the evidence that we have seen today into action. A lot of us are working at the front line, either in the facility, either in the lab, or even at the county level where you have your managing teams. Let us translate this information into action so that it can bring an improvement at our workplaces. We launched the WAAW just the other day. It is a whole week's commemoration. So this is just, we are the middle. Mine is to request that we continue the awareness efforts. We have a number of webinars that are still upcoming. Let us join and support the drive to spread the message and create awareness to the different stakeholders. In line with the theme for this year, let our choices that we make reflect the current action, because we have a big influence over a future generation. We need to protect the health and the livelihoods of those that are yet to come. As I close, I want to thank the sponsors for the WAAW. We have many of them. I think we have not been able to list all of them, but at your individual level, we really, really appreciate the support that you have extended to us so that we are able to hold the launch, the symposium, and many other activities, even targeting the counties. I know there are quite a number of counties that were not able to conduct some of the activities that they had planned, but we are hoping that going forward, the partners in the field will still be able to support you even as you implement your AMR activities at the facility, at the county level, and even at the level of the public. So mine is to encourage you to continue creating awareness and taking action in line with this year's theme of Act Now, Protect the Present and Secure the Future. Finally, to thank all of you for your patience, your active participation. I'm sure the questions that you asked have encouraged the presenters in whatever they're doing, and also to show their appreciation and recognition of the work that they have done. And also for your patience, every time when we start from the morning until evening. With those few remarks, mine is to wish all of you a very successful week, and even as those who are traveling back to your counties, safe travels as you go back to your facilities. Wish you all the best. Thank you.
Thank you very much, Dr. Emmanuel. So I think we are free to go for our lunch. Yes. So I'll request one of us to pray for us, then we can disperse. So tuombe. Okay, let's believe and pray in the name of the Father, and to the Son, and to the Holy Spirit. Amen. Our kind and everlasting Father, we thank you for the gift of life that are given unto us. We thank you for the gift of today, Father. Father, in the mighty name of Jesus, we want to thank you for protecting us throughout the process as we started our launch on Tuesday up to today's end of symposium, Lord. Thank you once again for granting each and every one of us journey mercies to and from home, Father. As we are going to take our lunch, Lord, may you bless our food, Lord. Father, for those people who will still be traveling back home, may you grant them journey mercies. All these I ask, believing and trusting unto your able name. Amen. May the grace of our Lord Jesus Christ, and the love of God, and the fellowship of the Holy Spirit be with us now and forevermore. Amen. In the name of the Father, the Son, and the Holy Spirit. Thank you.